Hello all. In this video, we will cover the passive and active properties of neuron. Myself Chilik and my friend Santosh will cover this lecture. I will start with a general introduction and then I will talk about passive properties of neuron. Then Santosh will take over and he will talk about the active properties of the neuron. So let's start. So we know that brain tissue consists mostly two kind of cell. One is neuron that you can see here. Neuronal cell can generate the electric impulse and can propagate that. The other kind of cell is the glial cells. This is some subcategories like astrocytes, oligodendrocytes, microglia, etc. The glial cells cannot produce an electric impulse, but it can help the neuron to propagate that impulse by providing myelin sheath. Also, the glial cells provide a supportive and a protective environment for the neurons. Any information that we get, like that can be sensory or motor, is an outcome of a electrical signaling between neurons. So how does the neuron generate that electrical signal, how it propagates that signal, and how it interacts with other neurons? What special machinery does a neuron have that sets it apart from other cells in the body? We'll try to understand this question today. Today we'll be covering these points. I'll talk about the passive ion channels, Nernst equation, GHK equation, the circuit representation of a neuron, parallel conductance model, passive propagation. Then Santosh will talk about the voltage-gated ion channels, the action potential, Hodgkin-Huxley model, and the action potential propagation. So let's start with the passive properties of a neuron. At first, we will try to understand what are ion channels and what is the electrochemical gradient. So let's think this is a cellular system. The left side is the intracellular site and the right side is the extracellular site. And in between there is the lipid bilayer. Now the lipid bilayer is not permeable to any ions, but it has small channels which let ions to move through it. The ion channels are in general selective, and in this case we'll think that the ion channels are only selective to the potassium ion. Now in the beginning, inside in the intracellular side there is 1 millimolar concentration of KCl, and the outside also has the same concentration of KCl. So there is no chemical gradient, and as well as there is no electrical gradient also. The net voltage is 0. Now we introduce 10 millimolar KCl in the intracellular site. That changed the chemical gradient only, but still the net voltage is 0, as the positive charge of K is counterbalanced by the negative charge of chloride ions. But because now the concentration of potassium is more in the inside of the cell and these channels are permeable to potassium ion will start moving from inside to outside down its chemical gradient. When potassium ion start moving out, the outside of the cell will start having more positive ion and inside of the cell will start having more negative ions. That will create a potential difference. Because of the electrical potential difference, that potential difference will start driving K plus ion inside the cell because inside is more negative. Like this, some potassium ion will keep moving out and some will keep moving in. And at some point, at some potential different, the number of potassium moving out will be exact as the number of potassium ion moving inside of the cell. So there will be no net exchange of the charges. This will be the equilibrium condition, and that calls the equilibrium potential for potassium ions. So we'll talk about it in more details in the next slide. So what we learn from this slide is uh, the cell intracellular and the extracellular side are separated by this lipid bilayer, which has ion channels. And different ions are present in the inside and outside of the cell. Ion can only move through these channels, and these channels are selective for ions. Mostly these channels are gated, and that can be ligand gated or voltage gated. But sometimes these channels also can be leaky. And also when we are talking about electrical activity of a cell, sodium and potassium channels are the most important channels for electrical activity of a cell. Okay. So in this slide, we are talking that potassium ion reached in its equilibrium potential. So like that, every ion has its own equilibrium potential, and we can calculate that from Nernst equation. So here Ex is the equilibrium potential of an ion in which there will be no net movement of the ion from outside to inside or the other way around. To calculate this, we need to know what is the outside concentration of that ion, what is the inside concentration, and what is the valency of that ion. R is the universal gas constant, T is the temperature in the Kelvin scale, and F is the Faraday's constant. This RT and F gives a value around 25 millivolt. And if we want to convert L into logarithm base 10, then we have to multiply it by 2.303. That gives a number of like 58 millivolt. So let's think that the potassium ion has an outside concentration of 20 millimolar and the inside concentration is 400 millimolar. Then by putting this value and solving this part, we get that the equilibrium potential of potassium ion is minus 75 millivolt. That means at this potential difference, there will be no net movement of potassium ion. Similarly, we can calculate the equilibrium potential for sodium ion as well, and which is plus 55 millivolt. Now that we understand equilibrium potential of a single ion, we can talk about the resting membrane potential of a cell. Resting membrane potential of a cell is the potential difference at which there is no net movement of charges. Let's think about a simple cell which has only potassium ion channels. So the cell is only permeable to potassium ions. Now the resting membrane potential of the cell will lies exactly at the equilibrium potential of the potassium. Like here it is denoting by EK. That is because at equilibrium potential of potassium there will be no net charge of potassium ion. Now if we add one more type of ion channel, that is like sodium ion channel, then this will start changing, and that is because the concentration of the sodium ion is more in the extracellular side than intracellular side, and also inside of the cell is more negative. So both chemical and electrical gradient will drive sodium ion to move inside the cell. Once sodium ions start moving inside the inside of the cell will start becoming more positive, and that will reduce the electrical driving force of potassium. So more potassium ion will start going out of the cell. This will continue, and at some point, the potential difference will be such that the number of potassium ion going out will be exactly counterbalanced by number of sodium ions coming in. So there is no net movement of charges, and that stage is achieved here. At this stage, the cell will be in its resting membrane potential. So, when a cell has both sodium and potassium ion channels, its resting membrane potential lies in between the equilibrium potential of potassium and sodium ions. Now we can calculate the resting membrane potential of a cell, but to do so, we need a new equation, as the last equation deal only with one ion at a time. So here we will be using the Goldman-Hodgkin-Kurtz equation, also known as GHK equation. And with this equation, we can calculate the membrane resting voltage when there is more than one ion channel is present. So this equation takes in account not only the outside and intracellular concentration of a single ion, but also the permeability of the ion channel. Here pK, PNA and PCl are the permeability of the potassium, sodium and the chloride ion respectively. Here also, like in this equation, we will be needing the extracellular and the intracellular concentration of potassium, sodium, and chloride ion. You can see for the chloride ion, it is intracellular concentration by extracellular concentration, and that is because chloride is a cation, which changes the direction of the chloride current. Now, this GHK equation also has limitation. This equation can only be applied when Vm or the potential difference is not changing. So, if we want to see how the potential difference is changing in some scenario when the permeabilities are changing and all, we need to consider the neuron as an electrical circuit. So again, let's start with a simple situation in which we'll talk only about the potassium ion channel first. The equilibrium potential of the potassium ion will work as a battery, and the ion channel will work as a conductor. So Gk is the total conductance of the potassium ion, and that can be explained by Nk into gamma k. So gamma k is the conductance of a single ion channel, and the Nk is the number of the ion channels that gives the total conductance. If we have more than one ion channels, like here sodium, potassium and chloride ion channels, these are just resistor and batteries in parallel. Then in a cell we have a couple of resistor in parallel, but that's not all. We also have a capacitor. This capacitor is coming from the lipid bilayer. And why is that? Because lipid bilayer is impermeable to any ion. Positive ion starts accumulating on one side of the lipid bilayer, and that attracts negative ion, which accumulates in the other side of the lipid bilayer. So that creates a capacitor. So with this resistor, batteries and capacitor, we have a full model of the cell. Now to calculate the potential difference of a cell, we'll be using a parallel conductance model. So here we have this circuit representation of a cell, and total current applied on this is a combination of the capacitive current and the ionic current. Again, the ionic current is a combination of individual ionic current, that is sodium current, potassium current and chloride current. Capacitive current can be determined by the membrane capacitance into the change in voltage with time, dVdt. Now at resting or steady state, there is no change in voltage. So capacitive current also will be zero. Also in resting state, there is no net movement of ions. So individual currents, Ia plus Ik plus ICL also will be zero. We can write the ionic current as the difference between membrane potential or Vm with the equilibrium potential of that ion multiplied by the conductance of that ion. So we can write Ina plus Ik plus ICL as this. If I solve this, we get Vm equal to Ena into Gna plus Ek into Gk plus ECL into GCL by Gna plus Gk plus GCL, where Ena is the equilibrium potential for sodium ion, Gna is the conductance of sodium ion, Ek is the equilibrium potential of potassium ion, Gk is conductance of the potassium ion channel, ECL is the equilibrium potential of chloride ion, and GCL is the conductance of the chloride ion. Now, I'll be talking about two important passive properties. The first one is, in a special uniform cell, given a steady impulse of current, how the membrane potential will change with time. We will make some assumptions here. The first one is the membrane potential is uniform at all points. Second one is membrane resistance or Rm is constant. And the third one is the cell is ohmic. We know that total amount of current applied in the cell is a combination of the capacitive current or IC and the ionic current or II. The capacitive current can be explained as membrane capacitance into change of membrane potential with time, that is C dVm dt. And the ionic current can be explained as membrane potential difference divided by membrane resistance. If we simplify this part, we get this. Here this component RMC or the membrane resistance into membrane capacitance is known as the time constant. Time constant simply means how much time is needed to change the membrane potential from its resting membrane potential to 63% of the maximum potential given the applied amount of current. Now, if the tau is bigger, it will take more time to reach the maximum potential difference. And that is because tau can be bigger both by increasing Rm or increasing Cm. If Rm is more, that means the resistance of the cell is more, which will reduce the conductance. If Cm is more, that means it will take more current to charge the capacitor. Both will result in a delay in reaching of the maximum potential difference. The change of membrane potential has two distinct phases. One is the rising phase and one is the decay phase. If we want to determine the potential difference Vt at any time point t during the rising phase, we need to use this equation. Here Vt or the potential difference at any time point t is equal to V infinity or the maximum potential difference given this current into 1 minus e to the power minus t divided by tau or the time constant. To determine the Vt or potential difference at time t in the decay phase, we need to use this equation. Now I will talk about the second properties of the passive neuron, which is how signal propagates along space. Let's think that this is a neuron and here the diameter of the neuron is same across the length. Now this is the point where the current is injected. So we will get maximum potential difference at this area. As we start moving away from this current injection site, the potential difference will start getting less and less. So 0 is the point where the current was injected and x is any point across the length of the neuron. To determine potential difference at any point Vx, we will use this formula, that is V0 into e to the power minus x by lambda. Lambda is the space constant which determines the spread of this signal. Intuitively, from the site of the current injection, the amount of distance need to travel for the signal to decay down 37% from the original membrane potential is known as lambda. Lambda can be explained by root over Rm by Rho plus Ri. Rm is the membrane resistance, Rho is the extracellular resistance, and Ri is the axial resistance. Now because the cell is in a big environment, we just consider that the Rho is equal to 0. That means lambda is equal to root over Rm by Ri. So if lambda is more, the signal will travel farther. But if lambda is less, it will decay down slower. You will understand this property better when you will do the assignment on the passive propagation. So, in summary, we can say that the cell is an electrical system. There are various ion channels and ions are involved in regulating this membrane potential. The membrane can be modeled as a RC circuit, and lastly, potential decays exponentially with distance. With this, my part is over, and now Santosh will explain the active properties of the neuron.
Now I'll be covering the active properties of a neuron. So before going ahead, we'll understand what we mean by active properties. When you say something is passive or active, so passive is like just lying stone. Just push it, it'll move for a little small distance and it'll stop. So there's no response to it. It just uh, uh, reacts to the input stimuli. There's no activity in that. So it's defined that the active responses or active properties are those which are dependent on selective changes in the parameters in the stimuli. So any property which comes about as an emergent phenomenon based on selective changes in the properties gets qualified as an active property. So one of the major ones is action potential. That we'll be studying in the upcoming slides uh, on how that's initiated and how it propagates and so on and so forth. And this is one of the major active property of the neuron. It helps in various sort of its functions. So let's start off. So the major component leading to the active properties of neuron are the voltage gated ion channels. The gating is a part which qualifies it as active. So in previous slides you might have encountered the passive leaky type of channels. So they just leak out ions. They may be selective with selective permeability to various ions, but they are not gated. They do not open up for a certain stimuli. I mean certain selective parameter. So they keep leaking out, but there is no other hidden latent parameter which is it dependent on. So this dependency of a latent parameter or the inherent selective dependency is what makes something active. So the gating part of ion channels is what uh, makes it makes the whole thing active. And there are two types of gated ion channels. One is ligand gated and voltage gated. Currently we will just limit to voltage gated ion channels. So these are the ion channels which open up only to a certain voltage inputs. Here we are showing two types of channels. These are the major ones that lead to many other phenomena of neurons. Those are inactivating sodium channel and non-inactivating potassium channel. And why inactivating and non-inactivating? We'll touch upon them in the upcoming slides. But just to give a brief overview, this activating or inactivating is uh, is relating to the internal mechanisms of how the channels are gated. So in the sodium channels, you have two sorts of gating mechanisms. One is activating part, which is the channel opening part, and the inactivating part, which is the small protrusion, the gate which closes up. So these two contribute, and because of the presence of this inactivating component in this channel, we term it as inactivating sodium channel. Whereas in the potassium channels, you just have one sort of an activating gate, which opens up at certain voltage. And in this particular example, voltage, that is membrane potential, the difference between the intracellular membrane potential with the difference between intracellular membrane potential and the extracellular membrane potential constitutes V in minus V out, the membrane potential. So when that is altered from minus 100 to 50 here, the both channels open up, but the way they open up and close is totally different, and that's guided by certain kinetics, which we'll touch upon soon. So that's how it is. So there are two types of channels that will be considering here, and these two major channels bring out another epiphenomenon, that is action potential. So it's an action. So there's an activity for a selective input, and the selective input is that the input perturbation, input injection of current or membrane potential change should cross a threshold. So the white line that's shown here is the threshold, and if the stimuli, either current stimuli or membrane potential fluctuation, doesn't cross the threshold, all the properties, all the functionality will be in the passive domain. But once it crosses this, we have enormous complicated functionality that's happening, which lead to this action potential. So we'll study one by one on how action potential is generated and how this waveform has come up. So that's interesting to note, that it's all just the interplay between these two voltage gated ion channels, sodium and potassium. We'll go through each phase briefly. The first phase is the resting potential. So in the resting phase, uh, there's no input stimulus, it's just resting. So in the resting state, the sodium channels. So yeah, just to mention it beforehand, uh, the sodium concentration on the outside is greater than the inside, intracellular, whereas it's opposite for potassium. Potassium concentration is higher inside the cell than outside. So that determines the flow, whether it's an influx or outflux, right? And if you see the structure of the ion channels, on the sodium ones you have two gates, and these two gates refer to inactivating. The down part is the inactivating part, and the upper one is the activating part. So inactivating part is one that closes up if you increase the potential, and that's the opposite. The activating part is the one which opens up when there's high potential. And in potassium channel you just have one gating, and this is an activating type, so it just opens up gradually. It's a delayed rectifier sort, so these gradually open up with increased depolarization. Yeah, there are two terms that we'll be using here: depolarization and repolarization. Depolarization is uh, increasing the membrane potential. So increasing, I mean, uh, change depolarizing, that is to increase the potential inside in comparison with outside, whereas repolarization is the opposite. So when the external membrane potential is highly polarized, where where it has high charge, positive charge in comparison to the inside. So these two terms just come up in the description. Okay, we'll start off. So in the resting state, does the inactivating is open because it's resting state. There's no much potential, so inactivating is open, but all other activating parts are closed, both potassium and sodium. And with an input stimuli which raises the membrane potential from resting to certain higher potential. So with that fluctuation stimulus, the sodium channels open up, the sodium activating part. So once these open up, because the gradient is from extracellular to intracellular, sodium channels start getting into the cell, and with that influx the membrane potential further rises up, right? So it's sort of positive feedback. Because of fluctuation, sodium ions come inside. Because of sodium ions coming in, the membrane potential rises still further. And again, because membrane potential is again rising further, the channels open up much more. So here just some of them. So this one is not yet open. Some of them open up. So with further increase in membrane potential, others also open up. That's a larger section of sodium channels that open up. So at certain point, most of the sodium channels open up, and there's so much influx. So influx leads to depolarization, increase in membrane potential, and that is this third phase, rising phase of action potential. So as a cascade, so many sodium channels open up and just get so activated, and it rises up. And at a certain membrane potential, the inactivating gate starts to kick in. So that's when it stops rising further. So that's when the falling phase starts. So inactivating part close up now. So inactivating part of sodium channels close up. Because of that, the influx, sodium influx, stops gradually. And so sodium influx was the main factor which led to increase in potential depolarization. Now when you close them up because of the inactivating part, and this is all voltage dependent, because you reached a higher membrane potential, the inactivating gates starts functioning and they close up. Now there's another concomitant influence of this, of increase in membrane potential. Not just the inactivating part of sodium channels close up, but the activating part of potassium channels open. So with the opening of potassium channels, as the gradient is from inside to outside, so potassium ions are more on the inside of the cell than outside. Because of this gradient, the potassium starts rushing outside. Now we are losing charge from the inside to outside. So what's going to happen? This will lead to reduction in membrane potential, repolarization. So that's what is this fourth phase, falling phase of the action potential. So this leads to further reduction in membrane potential, that is repolarization phase, and so it falls down. So uh, this is the main phase that brings down the membrane potential. Now how long will it keep going out? Unless uh, unless this either concentration gradient curbs down or the gate closes off, right? So what happens first? So what happens first is, one thing to note is that the concentration gradient is so large uh, that it doesn't happen soon. Okay. and the other part is that function the gating function is dependent on membrane potential, as we discussed earlier. And at a point, let's say around this point, the membrane potential is around resting potential. But uh, so practically the potassium channels should be closed off, because they were closed in the initial condition resting state, right? We saw here they were closed. But because the kinetics of the gating particle in the potassium channel is sluggish one, delayed one, these take time to close up. Because of that, potassium channels still move out, reducing the membrane potential further negative, and that leads to the undershoot refractory period. And in the undershoot, potassium channels are still on and they're just going out further, and so these channels take time to sense the change in membrane potential. That's why this happens. And after some time, these close off, and uh, the membrane potential uh, goes here, right? And then the upper part is because of the leakage. So after this, both sodium and potassium channels close off, but there are other type of channels, leaky channels, that we heard of, the passive type of channels, and because of them, the ions again come back to equilibrium. This is the whole phenomenon that happens behind the action potential generation. Okay, so the last part, undershoot, is what is referring to refractory period. It's the period after the action potential, uh, where the sodium channels are still on, but sorry, not sodium, potassium channels are still on, but not the sodium ones. So in this period, the sodium channels are still leaking out, and that's why the membrane potential is further repolarizing, goes down further negative. So what is the characteristic of the refractory period? So characteristic is that one cannot shoot another action potential during this period. So that can be appreciated by this simple experiment, and the experiment is to inject current at various time points with respect to the onset of action potential. At t equal to 0, if you just give a small current injection, you have a standard action potential, and you have this refractory period where the potassium channels are still on, whereas the sodium channels are closed. And just to give a theoretical understanding why it's hard to initiate another action potential, and that's because the sodium channels need further depolarization for initiation of the whole phenomenon for action potential, and that is counteracted by the opening of potassium channels. So potassium channels current is from inside to the outside of cell, and that is in a way inhibiting sodium channels to open up. So there's a lot more to it. You can read up the books to understand further, but to put it simply, in the refractory phase it's hard to initiate an action potential, and only after it comes down to resting membrane potential, after a while, can you initiate an action potential properly. So if you inject an stimulus, current stimulus at around 4.5 from 4.5 milliseconds from the onset, just a small pulse, small input. So this is not able to bring in more of sodium into the cell. So if you place it further apart, around 6 milliseconds, It's a bit better, and this is for 9 milliseconds. And if you place it further apart, it will look alike as it was in the initial part. This shows how action potential amplitude varies from the interval between the stimuli. This is just an experimental one, and this is a model part. Coming to Hawking-Huxley model. So this is a model proposed by these two eminent scientists, Hodgkin and Huxley, to understand and give a mechanistic interpretation of how sodium and potassium channels open up during the course of action potential, and what is the actual dependency of membrane potential on this uh, channel permeability and so forth. So they were the first ones to devise voltage clamping technique, and this is the technique to understand each channel permeability. So this is important because these are active channels, right? So consider there's a black box. You want to study its VI characteristics, right? If you have some electrical impedance, either maybe a capacitance, inductance, or some resistance, what we do is we put some voltage and see the current, and then try to interpret the components, impedance, capacitance, inductance, right? So similarly, if you try for the active part, the active iron voltage gated ion channel, it will not work because uh, if you put an input of some, let's say, some current, it fires an action potential, it just fires and goes off. You can't study it. And even if you put a small membrane potential which is uncontrolled, then it again gives rise to various currents. So there has to be an active mechanism, feedback mechanism to control the whole voltage, and that's how they came up with the voltage clamping technique. It's a whole lot complex technique where they get a feedback of current to change the voltage and so many things. So you can check out this technique in the book, reference book that we cited on the net, right? So they use it further to isolate sodium and potassium currents, and they theoretically explain the action potential waveform. And in fact, they were awarded Nobel Prize in 1963 for the work. Mainly, they worked on a certain axon called squid giant axon. So squids are a type of invertebrate where they have very large diameter axons. This is a ganglion from which you have stellate nerves of which in middle you have a giant axon, of which the diameter is around 1 mm, 800. It says exactly 800 micrometers. This is how it is. So you can remove that, study intracellular ion concentrations, and even put that in a bath, a salt bath, seawater, and study its membrane potential characteristics with input current and so forth. So they did a series of 5 experiments that they published in 5 papers that that explain action potential. To put it in short, uh, these are the 5 phenomenal papers. You can look it up. They did all these 4 in 1952. So we'll just touch upon the titles and what each paper covers. Their study explains these 5 studies uh, sections, overall coming up and proposing an analytical model of potassium and sodium channel permeabilities in the 5th paper. So the first paper is on devising this voltage clamping technique, and the second one is to trace apart the sodium and potassium current separately from the whole current waveform, and that they do skillfully uh, by tinkering with the external extracellular solution. So so they do it in this way. First, they place the giant axon in a solution where the sodium concentration on the outside is replaced with the choline. So with that, they interfere with the sodium ion gradient across the sodium ion channel side. So that in a way nulls out the initial part of sodium current, and just remaining current is of potassium current. So the potassium current that they get, if they subtract this from the total current, they get the sodium current, and that's how they trace apart these two currents for various membrane potential clamps, and trace out the current waveforms, and from them again they validate uh, whether the transient component is of sodium or not, whether the delayed component of the current waveform is potassium or not through some radioactive experiments. There's a whole lot involved. So we can check these papers. And the third paper is about the conductance waveforms. So from current you can compute conductance, and they trace apart the conductance waveforms, because conductance in a way is an indicative of membrane permeability, right? So as you might have touched upon in parallel conductance model, so the conductance is actually select permeability of the channel. So once you get the conductance waveforms for each of the channels, potassium and sodium channels, you can make up some models to reason out the variations. So they came up with various models to explain sodium and potassium channels conductance waveforms. And for the sodium one, they devised a dual effect, which is both activating and inactivating. As we discussed in the action potential, there is activating gate as well as inactive gate. So they propose that through some analytical studies. That's it. They try to come up with some exponential waveforms with either second order, third order, fourth order, and check it with the waveforms and see if it works. So that way they propose this dual effect, and that's really true, which is confirmed by the recent crystallographical studies. And the fifth study is the whole sum of all the previous ones, where they propose an analytical solution for the permeability of both inactivating and activating part of sodium and activating component of potassium and so forth. And the equations are this way. So yeah, conductance is for each of the channels GK, GNA. This is the leaky channel. This is the capacitance for lipid bilayer. So overall input membrane current is summation of all these, and each current is a function of the conductance, and they model this conductance, the permeability, as a function of uh, some latent variables. So these latent variables follow first order kinetics as highlighted here. Thus the simple first order kinetics, where dn by dt is function of alpha into 1 minus n, that is the output. So from n to 1 minus n, right? So it's like a radioactive disintegration, as you might have encountered in your physics. So similar sort of thing. So the channel is changing from inactivating to activating phase. So that's how they model it. And from this, if these are the rate equations, then you have the coefficients which give rise the alpha and beta, that give up your infinite value of this for n and infinity and tau. These define the activity. So what n infinity is the final n value that it reaches at t equal to infinity. So this is just a first order kinetics. So initial n is 0 and n infinity is n infinity, so it will be an exponential increasing curve. Similar with m, because these both are activating, and H is an inactivating, so it will be a decreasing, decaying exponential function. And here H starts from 1 and H infinity is this, so as it decays, and tau are the time constants for that variation. And again, coming to the conductance equations, so you see n power 4, this is the fourth order uh, function that they propose. So they propose n power 4 only based on mathematical fittings. So they have the experimental variations, and they try out various things to match that, and they propose n power 4. Same here, they tried m1 h square again m2 h power 4, various combinations, and they landed up at m cube h. These are the dependencies of the both n infinity and tau n. The steady state, the steady state, the active components of the channels, which are this inherent latent variables that we discussed, right? N, M, H for the rate kinetics. These are the n infinity dependency on membrane voltage, and the rate constants, time constants, dependence on membrane voltage. So these are so intricate that even if you shift this waveform slightly to the right, you won't get action potential. So that involved this is. You can even check in the advanced tutorials of neuron software. Uh, you can uh, shift this function here, uh, tilt it slightly, and move this here and there. Even slight modification deranges this action potential waveform. So such intricate is our nature. So they came up with such involved interactions between these channels that lead to an action potential. So it's remarkable to note that this highly complicated machinery is going on in us in our neurons. Isn't that great? And we found that our and these just show how potassium and sodium currents look like. Right, this is just n power 4, just like first order exponential increasing function. And this is the infinite value of potassium current dependency on membrane voltage, and this is the peak Na dependence on voltage. So this is monotonic, whereas this is not, because of the two gating variables that we discussed, M and H. One is activating and inactivating, and they play in such a way that they give rise to this. This is to put in short, like summary figure, which shows uh, how the sodium and potassium conductances vary during the action potential. So, so conductances are a label for permeability of that particular channel. So here the sodium channels open up, and because of this open up, uh, potassium channels also open up. So in a way, just to understand it properly, this is like a friend following another friend. So once the sodium channels start going in, potassium channels also start going behind it. They open up, but the closing part is so quick transient that uh, once some particular membrane potential is reached, the sodium channels just close up. But this friend, the potassium channel, is not as quick as sodium, so it takes time to come back. So it's like one of the friends getting uh, caught in ocean. So they too went into the river for swimming, and one of the friends came out quickly, but this couldn't, and so that leads to the refractory period, and it takes a lot of time because it's sluggish, it has such kinetics, and That's how it again comes back to resting membrane potential. Now we'll understand action potential propagation. That is to understand how the action potential that's generated at a particular location propagates across the axon. So once it's initiated by injecting some current, the sodium channels get activated, and then potassium channels, and uh, that affects the nearby potassium and sodium channels, and that in way propagates. That's what they explained here. So the first phase is that sodium channels open up, and that leads to action potential, and then uh, because of the change in membrane potential, that even affects the nearby sodium channels. So some depolarizing current flows. So the input ions that come in because of the action potential, that flow here and there on the left and right of this particular location, and that influences uh, the nearby sodium channels to further open up. So because of this depolarizing current that flows down the axon, right, that leads to local depolarization. And this depolarization open, as we understood earlier, so depolarization affects the sodium channels, affects in the sense it makes them more inclined to open up, and that go upstream and even inactivate. Yeah, this inactivation is of sodium channels in the upper part. Right. Yeah, this particular fourth part is of explaining why action potentials do not go all the way around. So if you initiate here, it will flow down on the both sides, but it will it will not exist at that particular moment. So it keeps moving. So why is this moving tendency there? Why it just doesn't uh, like pass on, doesn't flow down without change in here? So what I'm saying is, if you inject some water here, it should just flow down, keeping everything watery, right? But what currently happens with action potential is that once it comes here, the same thing goes forward, making the other part empty. So action potential moves because because of this refractory period. So because of the refractory period, the sodium channels close up, and that uh, inhibits that part of that location of the axon uh, from getting influenced by the sodium input, sodium currents, and the action potential. So that's why it just propagates in one direction. Yeah. Next, we touch upon solitary conduction. So our nature has come up with another amazing thing, amazing intelligence to increase the conduction velocity. So in the previous part, the sodium channels uh, open up gradually one by one, right? Whereas if you have some insulator, which is actually myelin sheath, which is made by oligodendrocytes, one part of glial cells. So if you insulate the part where you have where you don't leak the current, what happens is the sodium ions that come in because during the initiation of action potential at one location go directly without any leakage and influence the channels in the next of the node. So these are called nodes of Ranvier between the myelin sheath. So they influence quickly because there's no leakage. Because of that, uh, the sodium that comes in here again goes here, goes here. It's like jumping from one node to another node. So saltate actually means to leap. Yeah, saltate. It means to leap. So that's how, because of this jumping mechanism, the axons can conduct even more farther. So it increases the conduction velocity. So there are other consequences of myelin sheath, that is, it alters the membrane impedance by increasing the capacitance, and also affects connection velocity, that we understood. Just to summarize, what we have covered throughout is the voltage gated sodium and potassium channels and how they interact actively to generate action potentials, and repolarization phases of the action potential, and the Hodgkin-Huxley papers and their model of uh, the gatings to understand action potential and action potential propagation, how it's active and there's no attenuation. Yeah, this I didn't hint on there, highlight. That is the action potential waveform, the membrane potential, if you see, that doesn't deteriorate across space. So when the passive propagation was considered, the potential falls down with space. So it is just limited to a particular location. Whereas here it can propagate longer durations without any decay, and that's possible by myelination. So that's it, done. Just to hint on the tutorials, the first and second tutorials are these passive and active responses. So if you inject low current, then they're just passive, just like a RC circuit. Whereas if you inject more than threshold, that leads to action potentials. And this is like rate coding. The more the stimulus duration or amplitude, the larger that you have the number of action potentials. So this sort of rate coding. The amplitude of stimulus is coded in the number of spikes. And tutorial 3 is about refractive period, as we discussed in previous slide. And tutorial 4 is about passive propagation. And tutorial 5 is on active propagation. And you can see how the action potential waveform doesn't vary with location. Yeah, that's it.